Okay, so now we are on here at 20.5, and so this is all about some practical applications of manipulating DNA and genetic technology. So there's some medical applications. So one benefit of DNA technology is identifying human genes in which a mutation plays a role in a genetic disease. And so obviously we can sequence genes to see if they have the disease gene or not. Another way is this next thing, diagnosis of diseases. Scientists can diagnose many human genetic disorders by using PCR. So remember, that is amplifying many copies of a gene very quickly, or a segment of DNA. It doesn't have to be a gene. And primers corresponding to cloned disease genes, then sequencing the product to look for the disease-causing mutation. So you get many copies of it, you sequence it to get, remember, the order of the bases, and then compare it to a regular normal gene and look for that disease-causing mutation. So even when a disease gene has not been cloned, we can also still find it and see if somebody has a high likelihood of having that disease gene. So the presence of an abnormal allele can be diagnosed if it's closely linked to a RFLP. Remember, that is a restriction fragment length polymorphism, which means that there's slight differences between DNA sequences, and they call those markers, has been found. So they found links between disease-causing alleles and these restriction cutting sites, and so that's what we see here. So this is the disease-causing allele, and this is the normal allele. Notice here there's one cutting site associated with the normal allele, but two cutting sites associated with the disease-causing allele. So that is one way to actually look for, without sequencing, I guess, the disease-causing gene, to see if a person has these two markers, these two restriction sites, because if they do, then we found with much research that there's a very, very high probability that they also have the disease-causing gene, because they've found that with the normal allele for particular disorders, they may only have one cutting site. So if they find the two cutting sites, that tells them that most likely the person has that disease-causing allele. And the cutting sites are very close to the allele. Notice that they're not part of the allele. They're very close to the allele for the disease-causing gene, and therefore won't get mixed up with crossing over or anything like that, because remember, the closer that things are on the DNA, the less likelihood that crossing over is occurring. So therefore, whenever you have these sites, then the probability of having that disease-causing allele is high. All right, so that's another way besides just sequencing the DNA that they can tell, a little bit easier than sequencing the DNA, if you have the disease-causing allele or not, is these RFLPs. Okay, so now notice here, we're skipping over to chapter 21.2, and in the newer textbook for our book, they include this within this section, but our book separates them. So if you remember, we did chapter 21, but we skipped part of it. We did only certain sections of 21. This is the section that we skipped that we need to know about. And this section is about cloning organisms and also stem cell research. So we're going to talk about cloning first. And we're going to talk about it first with plants. So one experimental approach for testing genomic equivalence, meaning all cells of an organism have the same genes. We now know that that's the case, but there was research done to prove that, is to see whether a differentiated cell can generate a whole organism. So can you take remember, differentiated means it's already become specialized. So we started out being one cell big, but eventually as our cells divide, they become specialized. We have cells of our eyes, cells of our organs, skin cells, all kinds of different cells, and they become specialized and certain genes are turned on and off. So can we take an already differentiated cell that's already been specialized and kind of de-differentiate it so that it can develop into a whole organism? So in plants, they have cells that are called totipotent cells. A totipotent cell is one that can generate a complete new organism. So cloning is using one or more somatic cells. Remember, somatic, that's a body cell other than a gamete, from a multicellular organism to make a genetically identical individual. So can we take a multicellular organism, take a skin cell or a cell from the body, and use that cell to grow a new individual that's genetically identical to the individual you took the cell from? So they did this with plants first. So this picture is of your carrot. So the carrot, the part that we eat, so this is the green part up here. So the part that we eat is the root of the carrot. So this is the root, this is the carrot, and then there are roots coming off of there with root hairs and so on. Okay, so that's what we're looking at. So what they did is we look at this arrow here, they give you a cross section, a transverse section of the carrot root. So that's we cut it in half here. Cut it in half, this is what we're looking at. Then they chop it up. So now we have, instead of a section, we have chopped up pieces of carrot. Then they put it says, into a nutrient medium. So what you can't see, my picture doesn't show very well. Here's a beaker, and you have this nutrient medium. So the nutrients are energy sources, and you have the fragments in here, and then it says stirring causes single cells to shear off into the liquid. So you have little pieces, and then you agitate it and stir it up, and so cells on the very edge of those pieces will come off. So we have tiny, it's kind of hard to do tiny little cells in here, but there are also tiny little cells in there. Then what happens is these single cells begin to divide. So that's what we see here. So you have this, looks like it's 4 different cells. So 1 cell became 2 cells, and then 2 cells became 4 cells, and that's how we get this particular thing. So then it begins to divide again, continually divide by mitosis. So this is mitosis that's happening. And you start to get an embryonic plant that's developing. So the root is starting to come out and so on. So it continues to divide by mitosis, and you get the beginnings of a new carrot plant. You take that plant, and they cultured it on an agar medium. So here's the medium here. So the roots go down below here. All right, and then the green part goes up here. And then eventually, as it grows, transplant it into the soil. So if this is the soil, this is your new carrot plant. And this carrot plant, because it started out with a cell from this carrot plant is this is a clone of that, all right, that carrot plant. They're genetically identical. And so we took a totipotent cell. So the cells from plants are totipotent. You can take that carrot cell, a cell from that carrot, and it can develop into a whole new organism, which happens to be a carrot plant. And so that is a method of cloning. And so plant cells happen to have a lot of these totipotent cells. You may have unknowingly cloned a plant. Some plants you can take, this is the stem, these are the leaves. Anybody ever done this? So you have your plant. Anybody ever cut off a cutting of a plant? So you take off a part of the plant and put it in some water. So you have this cutting of a plant. And what happens is it'll begin to grow roots off here, and then you can transplant it into a pot with soil, and then that plant will begin, here's the roots, the plant will grow into a whole new plant. And so you can get, this plant would be a clone of this plant. All right, genetically identical. So that's another method there. This is a little bit different in that they're studying, instead of taking a whole part of this, they're taking a particular cell. And this was of interest because remember that we were trying to figure out, does every cell have all the DNA and so of the organism? And obviously yes, if you can take a cell from this carrot plant and it can develop into a whole new carrot plant, it must have all the genes that determine what a carrot plant is going to be. And so that was one of the purposes for that. So then, of course, we try to apply that to animals, and so what can happen to plants to animals. And so they did some what we call nuclear transplantation in animals. What they found is if you take a cell like this from from an animal, like from an existing animal, and then try and grow it, it doesn't really grow that well. It's not going to grow into a whole new organism, so we don't have that totipotent cell like a carrot plant. So these are some of the techniques that have been done to try and clone animals in a different way than what I just talked about with plants. So we do what's called nuclear transplantation in animals. In nuclear transplantation, the nucleus of an unfertilized egg cell or zygote is replaced with the nucleus of a differentiated egg cell. And experiments were done with frog embryos that shows a transplanted nucleus can support normal development of the egg, but we're going to see it depends on something. So if we look over here, here is your frog egg cell. So your frog egg cell here has DNA, it has a nucleus, and it has the DNA from the mom there, all right? And notice that they're applying UV light. UV light, what this does is it destroys the nucleus and the DNA. So of that egg cell. So that's what it says here. Then if we look here, it says enucleated egg cell. We've taken away that nucleus and therefore it's DNA. So you have this empty shell of an egg cell basically. All right, so then we've gone in two different directions, 1 and 2. So let's do this one first. So we go this way here. So here's that enucleated egg cell. So what we're going to do is replace that nucleus with the nucleus of a cell from an early development of a frog embryo. So here we have this other frog. It's not the same as the frog that we took the egg cell from. So we have this development of this frog embryo going on. We take one of those cells, which is very early on in development, so they're less differentiated. So this is not really a differentiated cell. It doesn't have a specialized job yet. And we take that nucleus from that cell and just extract that nucleus and inject it into this cell. So now this egg cell has a nucleus in it that, remember, has pairs of chromosomes, because this frog embryo started out being fertilized egg cell and then it starts to divide. And so let's say the frog has, let's say, 6 chromosomes. All right, so they have 6 chromosomes. That means this frog egg cell would have 3 chromosomes, right? Because it has half the number, and then we took out the chromosomes here. So this one over here, this frog embryo, all these cells are going to have the full 6 because it's going to have pairs of chromosomes. So now this egg cell feels like it's been fertilized because it has a diploid number. This has a diploid number of chromosomes. It has 2 of every kind because we took it from a cell that was a somatic cell. And so that's what we have here. So this has the same, it has a nucleus now and the chromosome number of a fertilized egg cell. So then it begins to divide by mitosis. Here's the early embryo. And it continues to divide, and notice here most develop into tadpoles. So it worked. All right, so most of it, not all of them, some of them didn't make it that far, but most of them did. So it seemed to work, like, wow, we can take this less differentiated cell, extract its nucleus, put it in an egg cell, and it actually does develop into a tadpole. Then on the flip side, the second direction here that we looked at is what's different about this one is the source of the donor nucleus. So now look at, they take the frog tadpole, take a fully differentiated cell. So that's what's different between this one and this one. The donor nucleus is from a cell that's now specialized versus not specialized. And so then we put that nucleus in that cell. So now it's a diploid cell, just like this side over here, and then it begins to divide by mitosis. Again, we get an embryo. But look at here, the stats. Less than 2% develop into tadpoles. So this was not very effective, but this was largely effective. What's the only difference between them? They took it from a fully differentiated cell versus a less differentiated cell. These cells are older. They've been the tadpole is older, so the cells have divided and been given time to fully differentiate. So it seems as if the age at which you take the donor nucleus makes a difference. And so the older they found the older the donor nucleus, so the older the donor nucleus, the results are not as good. Now, why would that be? Remember, when cells go through differentiation and turning on and off genes, we have methylation, we have acetyl groups added and turns on and off certain genes and so on. And so that's what they think is going on there. Okay, so this is the early stages of what we try to do with this nuclear transplantation in animals, is to clone, because really when this tadpole grows into a frog, and this will be an exact clone of the source of the donor nucleus, which is this frog embryo here. So those two would be clones of each other. This, though, if that's one of the tadpoles that is able to develop, will be a clone of that original tadpole. All right, so this is early stages of cloning. So let's move on with cloning, reproductive cloning of mammals. So you may have heard of Dolly the Sheep. In 1997, Scottish researchers announced the birth of Dolly, a lamb cloned from an adult sheep by nuclear transplantation from a differentiated mammary cell. So we did this actually with a mammal. So that experiment we just looked at was with a frog. So Dolly's premature death, though, this was in 97, and then she died in 2003. She had a liver disease and was actually put down because of it. And then she also had arthritis as well. Led to speculation that her cells were actually older. They were behaving older than those of a normal sheep of her actual age, possibly reflecting incomplete reprogramming of the original transplanted nucleus. So the nucleus, the DNA, was acting older from an older sheep than from a young sheep. So let's look at the process that we went through to clone Dolly here. So here we have the egg cell donor. So this is who donates the eggs. So we took her egg from our ovary, so that's what this arrow is showing you here. And now notice this little pipette, we're sucking out the nucleus. So take out the nucleus. Remember in the previous picture that was enucleated. This is an enucleated egg cell now. So then we want to put the diploid nucleus of the individual we want to clone into this egg. So that's her. So the mammary cell donor. So she's a different sheep. We took a mammary cell from a mammary gland here, cultured it. And what they did, notice here, it says cultured mammary cells are semi-starved. What that does is stop the cell cycle, and that's what triggers de-differentiation. That's important here. So these cells had differentiated into mammary cells, but they've done some research that if they take a differentiated cell and kind of semi-starve it to death, it causes them to de-differentiate and become more like the unspecialized cells, or more like from an embryo. And so then what they did is they took one of these de-differentiated cells and they fused them. So it's a little bit different method than the last picture we looked at. So they fuse them together, and now when they fuse together, the nucleus from this cell, so if this is the cell, this is the nucleus, we take that cell out, here's that nucleus, they fuse together, and so now the nucleus, which is diploid, because you took it from a somatic cell, you took it from the mammary gland. All right, so this is a diploid nucleus. So this egg cell now behaves as if it's a fertilized egg cell. It has a diploid number of chromosomes. The DNA is identical to the mammary cell donor. Begins to divide by mitosis, so you get your early embryo. And then as they divide by mitosis and get this early embryo, then they physically implanted that early embryo into the uterus of a third sheep. So we have sheep number 3 here. A third sheep. She has nothing to do with the DNA or the egg cell that came from these other 2 cells. She's going to be just the carrier. So she's the surrogate mother. And so they implant it into her, allow it to develop in the uterus into a sheep, and then they have the baby, and that's what we have here. We have the lamb that is born, and they named her Dolly, and she is identical to her. Because why? Because she is the one that donated the DNA. All right, so these guys are clones. They're genetically identical. And so, as I said, though, that this hasn't been wildly successful because of Dolly she acted as if she was older and had some problems. And so, like we wrote up here, that they tried to de-differentiate the cells up here, in that making them less specialized, but that didn't quite work out, I don't think, the way that they thought it would, and it really, at this point right here, really wasn't like a brand new fertilized egg cell that's new, like both DNA comes from the egg and the sperm cell. And so there's some issues there. So since 1997, they've continued work with cloning. They've cloned many different mammals, including mice, cats, cows, horses, and pigs. The first cat they titled Copycat. I've also heard them called Carbon Copy as well, was the first cat cloned. And so they've still working on the cloning process. All right, so the problems associated with this is that in most nuclear transplantation studies, only a small percentage of cloned embryos actually develop normally to birth. So going back to Dolly here, they did this process many times before actually getting a live birth of Dolly. So it's not like you just do it and it automatically happens. And also many epigenetic changes, such as acetylation of histones or methylation of DNA, must be reversed in the nucleus from a donor animal in order for genes to be expressed or repressed appropriately for early stages of development. That's what they were trying to do here with the de-differentiation and the semi-starvation, but obviously it didn't turn on and off all the genes that need to be on or off and de-differentiate it to the point where it's identical to a cell that has not been differentiated at all. And so there's some still issues there to work through.